Moving on from primary structure, we're now going to focus on secondary structure. The two major components of secondary structure are alpha helices and beta sheets. Secondary structure is the three-dimensional structure formed by hydrogen bonds between amino and carbonyl groups in the peptide backbone. So for example, here is our peptide, and we can see here's one amino acid, here's the next, here's a peptide bond, and here's our carbonyl. So if that is in three-dimensional space such that it interacts with the neighboring atoms from the same so let's say this peptide loops around. Here's another amino acid, here's the peptide bond, and here's the amine. So remembering that oxygen takes on a partial negative character, the hydrogen has a partial positive. So what that means is that these two groups here, if they get in the right geometry, they can form a hydrogen bond, stabilizing this geometry. Alpha helices and beta sheets are common examples of secondary structure, so we're going to take a close look at how those are formed and what stabilizes them. There are other secondary structure elements that exist. One example are beta turns. We will talk about those. There's also pi helices, other things. So if other things come up in your future, know that those are secondary structure elements you can look up. But for this class, we're just going to cover the major ones. Let's start with the alpha helix. So the alpha helix is held together by hydrogen bonding between backbone atoms. And most alpha helices are right-handed. So if you hold up your left and right hand in front of you and you kind of curl them in towards your body, you notice that they curl in different directions. So if you look at your right hand, you can see that that hand curls in the same direction that an alpha helix or most alpha helices curl. There are occasional left-handed alpha helices, but they're much less common. So let's take a closer look at that right-handed alpha helix. Here's a side view of it, and this is a combination of a ribbon diagram, which is that gray thick ribbon that you see, and then the atomic structure. So here you can see the atomic structure with the ribbon removed, and it's a little bit complex, but we can kind of follow. So here's the polypeptide chain. We see this carbonyl is reaching down and hydrogen bonding with this amine right here. It continues, we can see another carbonyl hydrogen bonding with an amine, and so it coils along like that. If we take that same alpha helix and we look down the top of it, you'll notice a couple things. One is that all of the side chains stick out around the outside of the alpha helix. And if we draw this in space fill model, so every atom occupies its real space in three-dimensional space, of course we don't know what the side chains are, but we can see that there's no space in the center of the alpha helix. So this geometry is just so that the atoms can just pack together with enough space. I think the best way to really understand the structure of the alpha helix is to be able to play with that structure in 3 dimensions. So we're going to do that in just a second. Some of the things I want you to keep an eye out for. 1, from the structure, notice that the side chains stick outside of the helix. Notice the location of individual amino acids in the helix. See if you can identify hydrogen bonds between the backbone amino and carboxyl groups. Sorry, I should say carboxyl groups. And consider how to interpret the ribbon model that is typically drawn. So oftentimes in proteins, we see an alpha helix like this, or we can also see just a cylinder. Both of these represent an alpha helix. So think about some of the ways that this representation is useful and some of the ways that it might be kind of limiting. So let's go take a look at that. Okay, so this is an interactive model. You can rotate it. You can use a scroll wheel or like a trackpad to zoom in and zoom out. If you get stuck or you don't know where you are, you can hit this refresh button and it'll take you back to the original orientation. So as I hover over it, a single amino acid will become highlighted. So we can see, starting at the bottom of the helix, here's one amino acid, here's the next one, and next one, etc. If I click on this, just a single click, it zooms in on that amino acid, and it shows me the position of the atoms there. So let's take a look. I can see, here's the backbone of the peptide, here's the side chain sticking outside of the amino acid, and if I look, I can see some of these hydrogen bonds stabilizing the alpha helix. So for example, there's a hydrogen bond between a carbonyl and an amine. If I keep looking, there's another hydrogen bond between a carbonyl and an amine. And again, we see all those side chains sticking outside of the helix. So I suggest to play around with this until the structure starts to make sense to you. It's a really nice interactive tool. In an alpha helix, they have this really specific repeating geometry because it is a helix. So we know that an amino acid, we're going to call it N for a second. Let's call this first one N, will form a hydrogen bond with the amino acid N plus 4 residues away. So this is N plus 1, 2, 3, 4. So that means this carbonyl is going to form a hydrogen bond with this amine. So what that means is there are about 4 amino acids per turn of alpha helix. Once I go all the way around the helix, it'll take me about 4 amino acids to do that. And then the next 4 amino acids will get me around another time. Okay, so those are alpha helices. Let's take a look at beta sheets now. So this whole thing that you see here in blue is a beta sheet. Each one of these arrows making it is called a beta strand. So if I'm talking about a beta strand, I mean just one of these. If I'm talking about a beta sheet, I mean the whole thing. The beta sheets are stabilized by backbone amino and carbonyl atoms in beta strands that hydrogen with the backbone atoms in a neighboring beta strand. So here is the same molecule, but now we can see the atoms, and the side chains are hidden in this view. So we can see here's a carbonyl, here's an amine, and they are hydrogen bonding. Go to the next amino acid, here's an amine, here's a carbonyl, and they're hydrogen bonding. So they're set up in just the right geometry that, again, those carbonyls and amines from the peptide backbone are lined up to hydrogen bond. You'll notice looking at this image here, there's very little space in between the beta strands. What that means is that the side chains don't fit in there. So typically the side chains are going to stick in and out of the plane of the beta sheet. So right now our beta sheet is in the plane of the slide. So you can imagine the side chains are going behind the slide and in front of the slide. In beta sheets, the side chains point in an alternating pattern. So for example, this picture is flipped 90 degrees, where now our carbonyl and amino groups point in and out of the plane of the slide. So it's 90 degrees flipped from the last one. So the carbonyl points back, the amino points towards us. And now the R groups, we can see the first one points down, the next one points up, the next one points down, the next one points up. So there's this very specific geometry that lines up these amino acids so that the backbone amino and carbonyl groups can hydrogen bond. Let's take a closer look at that beta sheet geometry. So again, we're going to use another interactive model. You can click on this hyperlink to follow along. Some things I want you to notice. Notice that the side chains stick above and below the plane of the beta sheet. Notice the location of individual amino acids in the beta sheet. Identify the hydrogen bonds stabilizing that beta sheet between the backbone atoms. And again, consider how to interpret that ribbon model that's typically drawn. What are the advantages and what are the disadvantages? All right, here's another interactive model. Again, you can use this button right here if you need to reset. So we can rotate this around. We can see, for example, here are 1, 2, 3, 4, 5 beta strands in a beta sheet. We can see that this beta sheet interacts with another beta sheet behind it. I think there's also 1, 2, 3, 4, 5 beta strands in that beta sheet as well. So again, you can hover over a certain place and each amino acid is highlighted. So I'm going to zoom in right here. All right, so now we can see the full atomic representation. So first thing we notice is that these side chains, again, stick above and below the plane of that beta sheet. And then if I turn in sideways, and maybe I can go, there we go, can I center? Nope. Let's try one more time. All right, it's a little tough to see, but you can make it out. So here is a carbonyl, and here is the amine it's hydrogen bonding with to stabilize the backbone. And if we move a little bit further along, let's check this one. There we go again. There is the carbonyl, and there's the amine that is stabilizing it. Here's the next one, carbonyl, amine. So I want you to take away from this that, again, what stabilizes two beta strands interacting with each other are the interactions between the backbone carbonyl and amine on the opposite strands. And then the side chains stick above and below the plane of that beta sheet. Beta sheets can have anti-parallel and parallel configurations. So on the left here, we've got our anti-parallel beta sheets. So in the ribbon diagrams, this is shown with one arrow pointing one direction and then the next arrow for the other beta strand pointing the other direction. So notice the way this geometry works is that your amine and your carbonyl are lined up pretty linear for those hydrogen bonds. That's in contrast to the parallel beta sheets where the arrows point in the same direction. And you'll notice it's not just the arrows, right? It's also the direction of the polypeptide chain. So this is going from the N to the C terminus, N to C terminus. They're pointing the same direction, versus an anti-parallel. On top we have C to N terminus, and on bottom we have N to C, so they're flipped. And in parallel beta sheets, you notice the geometry is a little different. So these hydrogen bonds are going more at an angle rather than the linear geometry seen in anti-parallel. Both can be fairly stable. You're going to see both in proteins. Just know that they do have different geometries. Okay, let's talk about beta turns. So a beta turn is the turnaround between beta strands in a beta sheet. So for example, here's a beta sheet, and we have a turnaround between this end of the beta strand to get to, oh, it's actually back there, to get to the next one. Or up here, we've got here, turns around and comes into here. So what you see in pink, those are beta turns. And if we take a closer look at the molecular structure, we can see this is what it looks like. So it's like four amino acids stabilized by a hydrogen bond between amino acid N, or I, and N plus 3. So here's N, N plus 1, 2, 3, and we see this hydrogen bond right there stabilizing that beta turn. The amino acids glycine and proline are going to be really common in beta turns. Glycine because it's a small side chain, so it can accommodate that tight geometry, and proline because we know proline induces turns in the backbone structure. So proline is a natural fit for beta turns. Let's talk about a couple special structures you'll see. So one is called a beta barrel. Beta barrels are pretty fun. They have this barrel shape that is made out of beta sheets. So the beta sheet goes all the way around, and the center of the protein is open, so things can flow through the center. So these are really common in membrane proteins. So here's another example from that side view, similar to this one. And then if we look at this protein top down, so I'm going to rotate it 90 degrees, we can see that it is a channel, so it's open in the center so things can flow through. This is one of the ways that the cell transports things across its plasma membrane. Another special structure is the coiled coil. So a coiled coil is when two alpha helices wrap around each other. So what's important is we have to have a lot of intermolecular contacts between these two coils, stabilizing this geometry. Another special structure is the collagen triple helix. So that helix is rich in glycine and proline. So when we look at the sequence, we can already tell it's going to be a collagen triple helix because we notice every third residue is a glycine, and we also have a lot of prolines in here. And then the third residue is a lot more flexible. So this is what the collagen triple helix looks like. So the molecular structure, it folds up like this, so we can see those prolines inducing that turn there, and then the glycines accommodating that small, the tight geometry there. Here's a space film model of it. So you can see these polypeptide chains are packed really close to each other. I want to be clear that the collagen triple helix is not an alpha helix. It's not three alpha helices coiled around each other. Instead, it's three polypeptide chains wrapped around each other and hydrogen bonded to each other. All right, so in your own words, describe what non-covalent interactions drive protein's secondary structure. Include a description of what groups participate in the secondary structure.